I developed one thermostable vaccine against Ranichet disease, and that particular technology has already been transferred to Asian #1 vaccine manufacturing company, Entry Biologicals, Venkateswar Hatchery, Pune. And after getting result from successful validation and also permission from Government of India, now it is under clinical trial, and the candidate is in IVRI Bareilly for further testing. And also recently reported one new virus in India, that is parrot borna virus serotype 4, that has been published in Nature Scientific Journal in the last month. And also reported one new virus, but today I cannot tell the name of that particular virus because I am still not getting the permission from Government of India. It is really causing havoc in poultry industry, especially in the broiler industry. Why I am saying these particular things? Because my research is based on KYC. I always believed on know your customer. Because without knowing our customers, we cannot think about the quality research and development. And already, before that, I want to say one thing. The State Animal Husbandry and Veterinary Department, Tripura, Dr. Tripura, very lucky because you are having respected Madam, Lipa Madam and Neeraj sir. They are having very good background of veterinary science. I heard from them and I am getting certain points rightly said by Madam that we have to work slightly different way. We are veterinarian. We are not simply a MBBS doctor. Right from epidemiologist, we are working breeding policy on this particular sector, therefore not getting the good germplasm also. Therefore, many things we have to do on this particular sector for survival of this particular sector and ultimately to grow our economy in terms of societal development. Now today's topic, that is from endemic CSF. Why I am saying endemic CSF, but actually I do not know about the Tripura, but CSF control program, literally it is successful. If we compare the reports of the classical swine fever virus for controlling any particular disease, first important part is understanding of the past. This particular important disease, classical swine fever, first reported in 1833. And after we are the superior brain, we are having the superior brain because we can talk and we can discuss. But viruses, that particular structure, only having the 3 components in certain only 2 components, genome and capsid only. That particular structural component can do devastating condition in the globe. Example is COVID-19. Therefore, this particular virus came into India in 2020. First outbreak reported in Assam in 2020. The differential points between the classical swine fever virus and African swine fever virus. If we do not know the chemistry of this particular virus, then it will be impossible to control the disease. This is the difference between 1950s or up to 1990s veterinarian's duty and today's veterinarian duty, because now we have to think about the chemistry, how the particular culprit organism can enter into a farm, pathogenesis, what are the tissue tropism mechanism, everything we have to know, otherwise we cannot control such type of emerging diseases, because before genotypes also, and no vaccine except in Vietnam. Therefore, another important point, if we compare these 2 viruses in terms of physico-chemical stability or environmental stability, then immediately we may say one thing, that African swine fever virus is more stable as compared to classical swine fever virus. And that is the biggest problem for us, and there is no vaccine. Ticks, ornithodoros, every transmittal routes are similar. In case of classical swine fever, direct contact, indirect contact, any infected material, anything. Another is domestic. In sylvatic, that is between the wild animals, and there will be major role of ornithodomestics also. But in case of domestic, from domestic to domestic and domestic to wild, in case of domestic wildlife interfaces, this is domestic life cycle and transmission. If we see the disease pattern as compared to classical swine fever, African swine fever is more acute. We may say part acute situation we may get. That means within a week we may get the mortality. But in case of classical swine fever, African swine fever, already I said, vaccine is like a life insurance policy. After purchasing that policy also I may die, but my family members will be survived. Into the farm, how that particular pathogen may spread from one infected animal to another one, then only we may develop one SOP. But now we are designing SOP on the basis of general viral disease control program. Therefore, and another important point, SOP, it should be a program. Like COVID-19, we have taken the mask. Now, therefore, biosecurity protocol, it should be a program, it should be a culture. It should not be like that. Today, African swine fever is happening, outbreak is happening in our locality. We have to maintain biosecurity. And another important point, to aware our veterinarian also, what type of sanitizer, what type of chemicals we will use as disinfectant. How that particular disinfectant will work. Because we are spraying certain disinfectant which cannot kill any microbes if we spray that. It should come into contact. These things, important things we have to monitor. Then on the basis of the locality, what is happening, we have to design the biosecurity. Biosecurity protocol of Tripura may not be same with biosecurity protocol of Assam state.